Before cell division, replication must occur. That is, the copying of the DNA should occur. Otherwise, the daughter cells cannot receive a complete set of identical complete set of DNA. So it must be ensured before the cell division that DNA gets copied and two exact copy go to each of the daughter cells. That's why DNA replication is needed. So what happens in DNA replication? In AS syllabus, this replication is simplified. Otherwise, this is very much complicated process, which you will understand or you will study in higher classes when you will pursue bachelor degree. But now, for now, we'll understand it in very simplistic way. Okay. So in the very first step, what happens? This DNA double helix unwinds and unzips as the hydrogen bonds between the bases, between the complementary bases break. And this breakdown is caused by an enzyme which is called helicase. In every step, there are different enzymes play their respective roles. So in this case, due to the activity of the enzyme, these hydrogen bonds between the nitrogenous bases, I mean the adenine, thymine, cytosine, guanine, between these bases, there are hydrogen bonds. So when replication starts, the hydrogen bonds between the bases are broken down. Okay. So after hydrogen bonds break down, now these complementary bases on each strand are exposed. Exposed for what? Exposed for getting new nucleotides. But before nucleotides are added to the complementary bases, these nucleotides must be activated. There are lots of this type of bases present in nucleoplasm of the nucleus. Okay, in nucleus there are lots of these bases, these nucleotides. But before they pairs before they pair with these bases, with their complementary bases, they must be activated. They must be activated by adding two extra phosphates. So when these extra phosphates are added to the nucleotides, they become activated, and these activated nucleotides can now base pairs. They can now base pair with the complementary bases on older strand. Okay, so what you can see, we see here that the nucleotides, activated nucleotides, are attaching, are pairing, are base pairing with the older strands complementary bases. Okay, if there is adenine, the complementary base is thymine. If there is cytosine, there is guanine. So this is the growing strand using this old strand as what should I say, template. This can be called as template. This is the template. Using the old strand as template, the complementary bases nucleotides are coming here, and the bond, the linking between the nucleotides are being carried out by the DNA polymerase enzyme. DNA polymerase enzyme does what? It links the sugar and innermost phosphate groups of the other nucleotide together. That is, this is a sugar, and this is a base. Here is phosphate, and this is another nucleotide, and there is also phosphate. So there is sugar. So the bond between phosphate and sugar is being made by the activity of this polymerase enzyme. Okay. So DNA polymerase enzyme. What is the role of DNA polymerase enzyme? If we can see it in detail. The phosphate is linking with the sugar by the activity of the DNA polymerase enzyme. Okay, these bonds are made by what enzyme? DNA
DNA polymerase
enzyme. But sometimes error happens in around 1 in every 10 to the power 8 base pairs. A few mistakes are made, and these mistakes are fixed by there are some enzymes, repairing enzymes, which repair or fix this problem in the gap 2 phase of the cell cycle. Okay, as we have seen in the last chapter during discussing the cell cycle, we saw that in gap 2 phase, replication occurs in S phase, and in gap 2 phase, replication errors are fixed. Okay, so there are 3 steps. In the first steps, unwinding and unzipping of the DNA double helix. In the second step, the activation of the nucleotides by adding 2 extra phosphates, and in the last phase, the activated nucleotides, pairing up of the activated nucleotides with its complementary base on each of the old strand, and a DNA polymerase enzyme is the enzyme which plays the key role here. That is, it links the nucleotide with another nucleotide by making covalent bonds. Okay, and this mode of replication is semi-conservative mode of replication, because this is the old strand and this is the new strand. DNA, there are 3 models suggested for DNA replication. So, see, this is the conservative model. This is the parental DNA, and in the resulting DNA, that is when DNA is replicated, you can see that one molecule of DNA is completely old, another is completely new. So a old DNA molecule is always conserved here. That is why this process is called conservative. But in this process, in semi-conservative process, what we can see that there is always a old strand and a new strand. By pairing between these old and new, these 2 DNA molecules are produced from this original DNA molecule. So what we can see that these DNA are hybrid DNA. One is old, another is new. That's why this model is called semi-conservative model. And this is the dispersive model. You can see that old DNA region and new DNA region, that is the parental DNA and the daughter DNA are distributed dispersely here. There is no absolutely old strand, absolutely new strand. Rather, there are there are blend of what? Old and new. Okay, this is called a dispersive model. This is called what? Dispersive model. But experiments suggest that DNA replication occurs in semi-conservative way. There, in the upcoming class, we will discuss how Stahl and Masselson prove this semi-conservative model of DNA replication. Okay.